Eye is considered as nature's most advanced camera. Like camera, it has two components. One is an optical component where it captures and focuses the image, and the neural component where it processes the image. So in our eye, cornea and the lens are the optical component. The retina converts this nerve impulse, send it to the optic nerve and to the brain. So this retina, more than an anatomical structure, it's a screen in which the entire world is projected. So every second what we see is being projected in this layer. So any pathology involving this layer makes the world dark. So before understanding the pathology, let's have an overview about anatomy, biochem and physiology of retina. So like any other structures, eye undergoes various processes during the fetal time. So at around 21 days of gestation, optic stalk with an optic vesicle formed from the cavity of the forebrain, where it grows in front of the lens placode. The lens placode invaginates this optic vesicle, converts them into a optic cup. So this optic cup has a two layer. One is an inner layer, another one is an outer layer. So the outer wall forms eventually retinal pigment epithelium and Bruch's membrane at around 4 to 5 weeks, whereas the inner wall forms outer neuroblastic layer where it differentiates into the rods and cones, horizontal cells and bipolar cells, and inner neuroblastic layer where it forms Muller cells, amacrine cells and ganglion cells. So this process takes around 12 to 28 weeks of gestation. So even macula undergoes the same process, but the remodeling occurs till 4 years of age, where there will be increase or decrease in the size of the diameter of the cones and continuous migration of the cone towards the foveal pit. So how to examine this retina? We have many instruments like direct ophthalmoscope, indirect ophthalmoscope. To get most advanced real-time 3D image, we have fundus photography, and to see the layers of retina, we have optical coherence tomography. So when we see through this instrument, actually what we see is a first one is an optic disc. The optic disc is nothing but the proximal part of the optic nerve. Then we have retinal vessels, retinal vein and the artery, retinal nerve fiber layer, then the macula. So the entire retina constitutes around 266 millimeter square, like a thin cloth, and it is extends from the optic disc to the ores aorta, and it is more thick at the optic nerve and thinner in the periphery. So this is the reason why when the eyeball expands, the more degenerations are more common in the peripheral retina, like in eye myope. So we have 10 layers of retina. Starting from the scleral side, the first one is a retinal pigment epithelium, then the photoreceptor layers, external limiting membrane, outer nuclear layer, outer plexiform layer, inner nuclear layer, inner plexiform layer, ganglion cell layer, nerve fiber layer, and the internal limiting membrane. So retinal pigment epithelium, this is an outermost layer, and it is firmly attached with the underlying Bruch's membrane and loosely arranged with the photoreceptors. The granular appearance of this is because of the unequal pigmentation of this layer. So the main function of this RP layer includes blood retinal barrier, where it pumps out the iron and the water for the photoreceptor renewal. For it also has phagocytic activity, and it gives mechanical support to the photoreceptors, like how we raise a concrete. The underlying earth is the Bruch's membrane, the overlying concrete is the RPE, and the support is the pillar is a photoreceptor. So same way it gives support to the photoreceptors. It also helps in manufacturing the pigment for the optical function. So coming to the layer of photoreceptors, we have two type of photoreceptors. One is a rod cell, where it is more in the periphery and it constitutes around 120 million. It helps in seeing in the dark. Whereas cones constitute around 60 millions. So they are more concentrated in the fovea, and it helps in the daylight and also for the color vision. So coming to the cones, again we have three type of cones, that is S cone, M cone and the L cone. Each of them will absorb different wavelength of the light. So to remember this remember these cones, I have a small mnemonic. So remember famous car names. The first one is a Land Rover, L cone for the red, then M cone, MG Hector, M cone for the green, then Suzuki Brezza, S cone for the B. So this is how you can remember this cones and what are the wavelength of the light they absorb. So when you see the ultra microscopically, so this photoreceptors has four parts. One is an outer segment where it contains various stacked disc-like structures where it deposits the pigment that is opsin. Then the inner segment where it is rich in mitochondria. Then nuclear layer that is a cell body. Then the synaptic terminal where it synapses with the bipolar cells. So this external limiting membrane separates the inner segment of this photoreceptors from the cell body and the synaptic terminal, that is a nuclear layer, outer nuclear layer. So the outer nuclear layer, as I already told you, is mainly formed by the nucleus of this photoreceptors. So this is where the place where there is a light detection and the signal transduction starts. So outer plexiform layer is nothing but where the photoreceptor synapses with the underlying bipolar cells. So coming to the inner nuclear layer, we have three important nuclear cells in this layer: horizontal cells, bipolar cells, and the amacrine cells. So the main function of this bipolar cells is to transform the information from the photoreceptors to the GCL. So the activity of this bipolar cell is being monitored by the horizontal cells. Then now the bipolar cells and the horizontal cell activity is being monitored by the amacrine cells. So the main function is it helps in processing and integrating the visual information. So again I'll repeat, the bipolar cells, it sends the information from photoreceptors to GCL, that is it gives a mechanical bridge between these two layers. The activity of the photoreceptor and bipolar cell monitored by the horizontal cell, the activity of bipolar cell and horizontal cells monitored by the amacrine cells. So then we have a inner plexiform layer where this inner nuclear synapses with the ganglion cell. The dendrites are arranged in this area. So the coming to the most important layer, ganglion cell layer. So there are around 0.7 to 1.5 million ganglion cells. So there are various type of ganglion cells like P cells, M cells, K cells, and there are few other cells like photosensitive cells. Remember these cells, I will explain in the later part of the slide. And there are some of the ganglion cells where it helps in supporting the circuits. So all these unmyelinated axons of this ganglion cells forms the nerve fiber layer. So the nerve fiber layer are arranged parallel to the surface of the retina. So depending upon from where it arises, for example, the fibers from the nasal side are called as superior and inferior radiating fibers. The fibers from the temporal sides are called as superior and inferior arcuate fibers. The fibers from the macula are called as a papillomacular bundle. So in case of glaucoma, superior arcuate and inferior arcuate fibers are the first one to get affected, whereas in case of non-glaucomatous optic neuropathy, this papillomacular bundle will get affected first. So this is how the nerve fiber layer are arranged around the disc. So the fibers coming from the deeper retina are arranged more superficial in the optic nerve, whereas the fibers coming from the superficial retina are arranged deeper in the optic nerve. So the last layer is an internal limiting membrane. It is a basement membrane. It is like a transparent sheet covering the other layers, like a transparent sheet covering the book, and this can be stained during the surgeries with the help of trypan blue, indocyanine and green, and it can be also peeled manually during the surgery. So the another important landmark of the retina is a macula. It is around 1.85 millimeter in diameter. So in the center part of the macula we have a depression called as an fovea. In this layer only cones with the overlaying internal limiting membrane is there. The other layers are absent. So in the center of this fovea there is a tiny depression that is called as an umbo, that corresponds to the foveal reflex. 0.5 millimeter around this foveal region or corresponds to the parafoveal area, and 1.5 millimeter around this corresponds to the perifoveal area. So another one is a foveal avascular zone. This is area inside the fovea but outside the fovea. It is devoid of the blood vessels. So enlargement of the foveal avascular zone indicates that there is a capillary non-perfusion seen in diabetic macular edema. Then as I told you, the retina extends from the optic disc to the ora serrata. It is the area where the retina ends and the ciliary body begins, and it is more wide temporally, and it is nothing but it looks like a serrated edges. So coming to the blood supply of retina. So retina is supplied by ophthalmic artery, but it's a branch of internal carotid artery. It gives a branch central retinal artery where it penetrates the optic nerve and enters the retina. The other branches are long posterior ciliary artery where it anastomosis anteriorly, and short posterior ciliary artery where it supplies the RP and then choroid. So let's see in detail. So central retinal artery, it penetrates the optic nerve, enters the retina through this optic disc. So as soon as it enters the optic disc, it gives superior, inferior, nasal and the temporal branches. So this central retinal artery supplies a inner layer of the retina, whereas this posterior ciliary artery supplies the outer layer, that is RPE, photoreceptors, RPE and the underlying choroid. So, in case of central retinal artery occlusion, where the inner layer of the retina is compromised, so the inner layer will appear opaque, whereas the outer layer still have the supply by the posterior ciliary artery, where it is see-through at the fovea, which appears like a cherry red spot. But in case of ophthalmic artery occlusion, where the entire artery is occluded, so both the central retinal artery and posterior ciliary artery blood is compromised, so the entire retina will appear pale. Then we have another one variant, that is a cilioretinal artery, which is seen in around 15 to 30%, and again the presence of this helps in vision saving in case of central retinal artery occlusion. That is, it prevents severe vision loss in the central retinal artery occlusion. So coming to the venous drainage, let's make it very simple. So along with the central branch retinal arteries, the branch retinal vein enters the optic disc and it forms the central retinal vein, where it drains into the cavernous sinus. So again we have another vein called, that is the vortex vein, where it drains the sclera and the other part of the choroid, where the superior it constitutes around 4 to 5 in number. The superior part drains into superior ophthalmic vein, the inferior part drains into inferior ophthalmic vein, and eventually drains into the cavernous sinus.